My name is Alex Ogbonna from Nigeria and a first-year PhD student in the field of plant breeding and genetics. Growing up, I had always thought of how I can be relevant to my community and the world in general. One of the best ways I can do this is to support smallholder farmers in sub-Saharan Africa. And this translates into being part of a larger effort to solve problems of food security in the world. Plant breeding offers me the platform to achieve my dream. So plant breeding is simply the genetic improvement of plants for human benefits. One of the staple food crops in Nigeria is yams. And so I want to describe a project that aimed at increasing yield and reducing the number of years it takes to release a new variety of yam in Nigeria. Previously, the conventional scheme that is being used that takes about 9 years before a new variety can be made available to farmers. So yam is a tropical tuber crop. It is vegetatively propagated, and millions in Africa depend on it for food every day. And yams are grown on over 5 million hectares in about 47 countries of the world, with Nigeria as a leading world producer. Now, specifically, white yam, which is the Ascoria rotundata, is the most widely eaten and grown yam species in the world. So it's very important, but outside of Africa. So major constraint to yam production in Nigeria, my focus will be in Nigeria. So one of it is that land races grown by farmers are characterized by low-yielding tubers. Tubers are generally susceptible to yam mosaic virus disease. Tuber quality and yield are reduced by up to 80% by pests and diseases. This is how much this constraint goes to reduce the yield that farmers produce in their farm. One other thing is it takes a long breeding cycle of about 9 years to release a new variety. So my research objective would be to develop a yam variety with high-yielding tubers and to develop yam cultivars that are resistant to this yam mosaic virus that reduces yield to up to 80%. And the last is to reduce the long yam breeding cycle currently used by the breeding program. So I propose a marker-assisted recurrent selection scheme that will shorten the breeding cycle while developing high-yielding, disease-resistant quality tubers. So this study will employ a recurrent selection breeding technique, which involves repeated cycle of selection and genetic recombination in order to improve agronomic characteristics such as tuber yield, tuber oxidation, flesh color, starch content, and resistance to yam mosaic virus disease in the population. Now, after the initial step of population improvement, superior materials and with trait of interest will be crossed and screened using markers and further validated through advanced trials, and then high-yielding and disease-resistant cultivars are released to the farmers at a shorter time period. I think younger people that are looking forward to helping their community and others, especially the smallholder farmers, plant breeding presents a good opportunity and platform for you to achieve this goal.